You all. Thank you, Dave Wood and Henry Heiser were really kind enough to invite me. They both are on our board. Half of our board is standing in front of me. This board really does drive what happens at the Brown Cancer Center. Without the community support that we get from them, we wouldn't exist. So I'd just like to take a second and applaud your actions over the past 10 years. So I always go over when I get caught, so I'm very careful not to do that because I know you have to be somewhere at 1 o'clock. And I thought what we could do today is really just talk about what the Brown Cancer Center is all about, what our mission is, what we do, and where we're headed. And so everybody knows that what Kentucky's known for. We're number one in the United States for college basketball. We are the number one state. Okay, let's go back. Okay. But you may not know that we're also number one in the United States for cancer-related deaths. And you can see that number one sign right there in the middle of Kentucky. And this is from the National Center of Institutes, Cancer Institute's database. And what they did was look at the number of cancer-related deaths that occurred over a 5-year period. And the red states are the states that have the greatest number of deaths. And if you look at that range, I don't have a pointer, but you'll see the orange Kentucky on the right side of the screen. That number at the top end of the range is actually Kentucky. So we have a huge health problem in Kentucky. We are literally ground zero for cancer in the country. And we need to really kind of step up to the plate and fight this right here at home. Cancer deaths in 2013, these are the anticipated deaths. It's based on trend lines and based on last year's numbers. In Kentucky, we're looking at about 10,000 individuals dying of cancer. And I would believe that each one of you has had a friend or family member die of cancer probably in the last 5 to 10 years. In the United States, it's just over 500,000 people who die every year, and it's expected 580,000 will die in 2013. What's interesting about cancer, unlike cardiovascular disease and some of the neurological diseases that tend to preferentially affect 70-year-olds, 80-year-olds, cancer really hits individuals at their moment of greatest productivity. In fact, I run a melanoma clinic and routinely see individuals in their 20s, 30s, and 40s with metastatic melanoma who have a lifespan that's measured in months as opposed to years. And you can see on this column graph here, the 18.3% of individuals who are 55 to 64. This is peak years of productivity. In fact, what I'm seeing with my boss, Don Miller, that peak years of productivity are really 65 to 74 now. And so we're really hitting the majority of our population when they're at their best and when they're most needed. Globally, it's an incredible problem. 8 million people will die in 2013 from cancer. And that translates actually to 15 deaths per minute. So I've been talking for about a minute while this slide has been up in front of you. And in that time, 15 humans died of cancer. So it's a huge problem. And Kentucky developed the Regional Cancer Center Board was formed in the late 70s and moved to develop a cancer center whose focus really was about providing excellent cancer care in Louisville and the surrounding areas. That's what the initial focus was. And when Dr. Biller got here, it changed a little bit because what he saw was the opportunity for us not just to be a great cancer center to take care of Kentuckians, but also to be a great cancer center that might be able to introduce new cures, new treatments, and build us not just from a community cancer center, but through to a regional and nationally recognized cancer center. So our mission essentially is to cure cancer. We want to reduce morbidity and cancer-related deaths. The second mission, which is essentially almost as important because they're going to continue the battle, is to train the next generation of cancer scientists and clinicians. And then last, and as was the case in the 1970s, we want to provide the most advanced cancer therapeutics and diagnostics that anyone can get in the entire region, so you don't have to get on a plane and go to my alma mater, Sloan Kettering, every time you get a CAT scan. So what's the Cancer Center really all about? We're not a department at the University of Louisville, actually. We're sort of a matrix organization. And essentially, the medical oncologist, I see patients, inpatients, and I see patients at Norton, the University Hospital, VA Hospital, Jewish Hospital. We really work across all the hospitals to take care of cancer patients. The scientists, some of the scientists that we have actually work in Owensboro, Kentucky, who are part of our center. So we're a matrix organization that crosses multiple entities, including the University of Louisville, which is our main base organization. What do we do? Well, it's really two areas. One is on the research end, which is the blue side here, and there we do basic laboratory research, and most of this research is done in that building. It's called the Clinical and Translational Research Building over on Hancock Street. And then we move basic discoveries into drugs, and then hopefully into clinical trials. And I'll show you a couple examples of that in a few moments. At the same time, as we're doing these studies, mostly in the lab and with mice and clinical trials, we're also trying to take care of the population. And the population isn't just Louisville, it's not just Kentucky. We are a major referral center for southern Indiana. We have patients that are sent to us from Vanderbilt, from Ohio State, from Duke University, depending on what trials we have available. And we strive to really generate enough clinical trials and enough clinical expertise and diagnostic imaging potential and therapeutic expertise to be a place where you can really feel at home and feel comfortable that you're going to be able to get high quality cancer care. And all of this plays into the education with the university, because we're educating those scientists and physicians as we do that. So I just thought I'd briefly show three quick examples of some of the projects to give you a sense of what we're all about. And this is a study that's run by Getz Cloker. He's the head of our thoracic oncology unit, so he takes care of lung cancer patients. And that graph there shows you the life expectancy of a patient who develops lung cancer. And you can see there's a precipitous drop in the survival over the course of 1 to 2 years. So the vast majority of his patients, when he sees them, will die of their disease. So he's really pushing for early detection, trying to detect the cancer before it spreads. And so he's working with a variety of basic scientists to identify tests that we can perform on blood as opposed to doing CAT scans every 3 months and getting the dose of radiation that is unhealthy, to do it on blood and see if we can detect lung cancer early, because virtually all cancers are curable if caught early enough. It's just a small tumor in your lung is a very difficult thing for us to detect. And so he's got a variety of projects. This is actually a short list of them, but I wanted to just briefly mention one of them. The highlighted one is plasma calorimetry, and this is a collaboration between Getz, who's a medical oncologist, he's a physician, and a biophysicist, a guy who actually studies molecules and the physics of how things change over time. And these are the two individuals, Getz Cloker, and the biophysicist is Brad Shares there. And what they did, they came up with this kind of crazy idea, which was if they looked in the plasma of patients, they might be able to see sort of a global effect on the ability of that plasma to change when you heated it up. And that concept is pretty simple. It's like taking a hot water heater and putting your blood in it, heating it up, and then seeing how much heat gets released. And that's dependent on the proteins that are in your blood and how they interact. It's way too complicated for me to understand, let alone to explain. But what isn't complicated is the thermogram that you're looking at. So the black line is what your thermogram likely looks like. It's that big peak that happens early in the temperature curve. All the other colored lines actually are different types of cancer. They're blood taken from different patients. So he has basically 100% ability to discern a normal patient's blood from a cancer patient's blood. So I saw this and was very skeptical. It's my job to be skeptical as a scientist, and decided that I would do a blinded test. So I gave him a bunch of normal samples and a bunch of samples for my patients. I take care of advanced melanoma patients, and asked them to tell me which ones were the cancer patients. And sure enough, they were able to tell me 100% of the time. And so all the red lines are the normal healthies in this graph, and then all the black lines are my patients. And there's about 20 different patients' sera. So this has actually led to a clinical trial. It's a multi-center clinical trial where we're prospectively following patients and doing CAT scans or PET scans every 3 months and then taking their blood at the same time. And we're going to correlate whether they have cancer with the CAT scan and whether they have cancer with this new test. And it's a good example of a collaboration that's occurring there where it's a biophysicist and a clinician. And I should have mentioned that there's a postdoctoral fellow, a trainee who's been working at the bench doing all this work. A second brief example is one that I've personally been involved in. It's been a pretty exciting journey. It started when I was at Sloan Kettering and Cornell, and Dr. Miller, when I was recruited, actually, I carried mouse embryos with me when I flew into Louisville, and he gave me my first grant, $14,000 to implant those into pseudopregnant mice to produce mice that didn't have a particular enzyme I was interested in. this project actually is all based on the concept that patients' tumors take up a lot of glucose. So if you get a PET scan, read them on billboards, all that is I'm injecting a labeled glucose into your bloodstream and looking to see where it goes. And in the case of this patient that you're looking at, you can see his brain at the top takes up some of the glucose, that gray circle at the top, and then way at the bottom of his body is another dark circle. That's his bladder, where he's releasing the sugar into the urine. And all those other black spots are all individual tumors. You can see those tumors are taking up a lot more. It's about 10-fold more sugar than the normal tissues. And so we identified an enzyme that actually drives this process, that regulates this process. It's sort of a switch that turns on the ability of the cell to eat sugar. And when we got here in 2002, 2003, we entered into a collaboration with an organic chemist named John Trent, who made a model of our enzyme. We did computational screening. He used computers to screen for these small little compounds. And he found one that looked pretty good to us. And we tested it in the lab, and it turned out it inhibited the enzyme. And it's shown there. It's got a long name. We call it 3PO for short. And it's a small molecule. It's got good pharmacokinetics, and it's a druggable agent. But it didn't have appropriate intellectual property. There were some issues with it. The potency wasn't very good. But we were able to show that it would block tumor growth. And this is a breast cancer model. The guy in the right corner there is Brian Crank. He graduated from Center College. He's a Louisvillian, was born here, bred here, went to the University of Louisville Graduate School for Biochemistry, then did his postdoctoral fellowship with me, and that's when he did this breast cancer model. These animals have a human oncogene inserted into their genome. And you can see that the animals that got the drug, which are the circles at the bottom line, the tumors grew at a much slower rate than the animals that did not. And Brian's now become an assistant professor and got his own funding from the National Cancer Institute and the American Cancer Society. Dr. Miller started very early trying to figure out ways that we could move these technologies into the clinic, and worked with the university and with Randy Riggs, who's here today, to develop a new concept company at Advanced Cancer Therapeutics, which is really a joint enterprise between the University of Louisville and private investors. And what they did was they licensed various compounds from investigators, cancer researchers, and they picked mine, it turned out, as one of their lead compounds. And then they helped us to generate more potent derivatives. And they actually had it done in India, where they would rationally design new compounds, and then they would send them to India to be synthesized, and then ship them back here, and we would test them in our lab. And this led to multiple iterations. A couple hundred compounds actually got tested, and eventually we found a compound that was about 100-fold more potent than the original parent compound. And that compound is shown here, called PFK15. And the little line graph there shows the 100-fold increase in potency because the line got shifted by about 100-fold. And down on the right is Julie O'Neill, a postdoctoral fellow who came from the University of Edinburgh to work here when she found out about some of the translational research that we've been doing. This compound's pretty cool because you can take a mouse and do a PET scan. And in the case of our mouse scanner, we do this down in the basement of that research building. The red is the glucose uptake. So this is a cross-section of a mouse with a large lung tumor on its flank there, and that's the big red thing where the yellow crossbars come together. And when we give the drug, you see the redness all goes away. So it blocks sugar uptake completely, and it also blocks the growth of tumors in a variety of different cancer types. This is just an example of colon cancer. The drug that's currently FDA approved, and one of the most potent drugs that we use for colon cancer is irinotecan, and this drug is actually a little bit more powerful than that. And so this has all led to a phase 1 clinical trial. It's not over here and at Georgetown University. All the technology and the funding is coming from the University of Louisville and from Advanced Cancer Therapeutics. The IND toxicity studies have been completed. The Food and Drug Administration is in the process of giving us the final go. And this is Rebecca Redmond, who's the principal investigator. She was trained at Duke University and recruited to run our phase 1 trials program. Then the last brief example I'm going to mention is an area, it's a clinical trial that actually started with a medical student, and they're really all about education. It started with a woman named Marianne Raskin. And there's Marianne at the Kentucky Derby. In 2008, she was in my lab tinkering about, and we were talking about different things that she could work on. And there's a drug that we use for lymphoma, and we thought that this particular drug might have an effect on a certain type of white blood cell that turns off the immune system. And so we tried it in the lab, and sure enough, it had this effect. It turned off this suppressor cell, which might allow the immune system, the body's own immune system, to fight the cancer. And that's really where a lot of cancer therapeutics are headed, is towards immunotherapies. And so we tried it in a few patients, and I don't have a pointer, but the upper right graph there has a bunch of little green and blue dots. And if you go to the graph right below it, there are fewer of them. And that's in an actual patient. We gave the drug to a patient, and we were able to show that it got rid of these suppressor cells. And so this particular patient had a bad disease, a horrible prognosis measured in months. And you can see on the far left there, he had very little cancer. Most of what you're seeing there is just normal physiologic uptake. He did have a few small subcutaneous nodules, but then over the course of 6 months, he developed all those black dots in his lungs and his liver. He got this agent that came from the University of Louisville, really from me working with a medical student, and had a dramatic response. He went from basically being at death's door to being able to lead a normal life. And what was interesting was after another 6 months, his tumors continued to go away. He was left with just one little tumor right in the middle there. You can see that black dot right in the middle of him that was left. And I had Chuck Scott, one of our surgical oncologists, cut it out, which turned out to be fortuitous for the patient, of course, but also for us because we got to look at the tumor. And all those little brown spots in this tumor are your immune cells invading the tumor and keeping it at bay. So we really proved that the concept that this drug could do this, which was pretty exciting for us. We've now done a 69 patient clinical trial. Here's just a few quick examples, a bunch of black dots, bad cancer, going away with just one cycle of the drug. Another individual with a tumor in his left lung and several right armpit tumors went away completely. A patient who couldn't even walk across the room, he had so much extent of disease in his liver. Didn't cure him, but you can see what it did for him over the course of, this is only 15 months, but he's lived now for 3 years. And then this is a great example I like to show. This is just outside our infusion area. This is a Greek admiral. I won't use his name, although I know he wouldn't mind if I did use his name. He's retired, of course. He flew in from Athens, Greece, to get on this clinical trial. And there were no other, this was at a time 3 years ago when there were very few options for him. And he flew in and was treated at the Brown Cancer Center on a clinical trial that we developed. And he had a complete response, and he's alive and well today, cruising around in his boat and constantly inviting Dr. Miller and I to come out and visit him. So the end of this last and final example is, the end of the story is that this trial that was developed here using philanthropic funds predominantly, a little bit of state money as well, but mostly philanthropic funds, was developed here. The concept came from here. It was run here. It is now a multi-center clinical trial across multiple states, MD Anderson, UCSF, you name it, in stage 4 melanoma. It's a sponsored trial, which means that a company is now spending about $15 million. We're the lead site. I'm the principal investigator on it. And it's the kind of story, it's the kind of progress that we want to make right here, that we believe we can make right here with really not that much more support than we're getting already from the state university and from philanthropy. So how are we going to cure cancer, and how are we going to move forward? I mentioned this in my little write-up, and I'm going to say something, and I'm hoping no one has any tomatoes in the room. And I should say that I haven't talked with President Ramsey about this, I haven't talked with the mayor about this, I haven't talked with anyone about this, perhaps except Dr. Miller, who said I'm crazy. But I got online and started looking at other municipalities, and it turns out that there's quite a few out there that tax cigarettes. And Kentucky doesn't tax their cigarettes much. It's about 60 cents. We're about the 40th lowest taxing state in the country. We actually get a ½ cent of every pack sold in the state of Kentucky, and UK also gets a ½ cent to support our program. That translates to about $1.5 million. What would a 5-cent tax on cigarettes just sold in Louisville do for us? Well, it would generate about $5 million. And it wouldn't quite double our budget, but it would increase it by about 50%. And it would mean we'd have more medical oncologists, more technologists, more cancer researchers. It would enable us to do what we're already doing, but do it in a grander way. And so the immediate concern I've heard about this is, well, we're an anti-tax state. we're anti-tax city. We can't tax people who smoke because they tend to be people of lower income. 5 cents on a $4 pack of cigarettes is not going to have, I think, any effect on the ability of most individuals to smoke. We're not talking about New York that's taxing $4. We're talking about 5 cents. There are 450 local jurisdictions that currently tax their cigarettes. That includes Chicago and New York, about 20 cities in Virginia alone, believe it or not. The most recent example of this is Omaha. Omaha put a 35-cent tax on their city's cigarette tax only for the cancer center, to fund their cancer center to get NCI designation. It's translating to about $35 million over a 10-year period. The only people in Omaha that cared, it wasn't the smokers, it wasn't the Rotarians, it wasn't the conservatives, it wasn't Republicans, Democrats, it was the state government. The state government said, wait a minute, we tax cigarettes. You don't tax cigarettes, we want to deal with that. So they got mad. It was a little bit controversial, but they got it passed pretty easily. The people who are taxed the 5 cents, they're the ones who are going to get the greatest benefit from it. Lung cancer, lung cancer makes up about 40% of all the deaths that I was talking about. Now, I should say 15 to 20% of lung cancer deaths, those patients don't even smoke. They've never been exposed to smoke. But the majority smoke, and the majority of the patients the individuals who would be taxed this would have the benefit of having really a state-of-the-art, highly innovative cancer center, not just the research going on, but the multidisciplinary care that's going on, and all the imaging that goes with that, and all the therapeutics that goes with it. And then there are these indirect economic benefits. And these are benefits not just with a cancer center, these are benefits that happen from having a university. And but for the cancer center, there's no doubt that we're bringing in right now around $20 million a year in peer-reviewed grant money. That's coming from the NIH, it's coming from the Department of Defense, it's coming directly through the U.S. Congress. This is money that's being brought in to the community and being spent right here, $20 million. So there's a huge possibility for this to lead to more of those. We believe this will lead not just to more of those grants being generated locally, but us to be able to recruit individuals to move here with those grants and spend them right here in Louisville. And then, of course, as we build our clinical reputation, we'll be getting more and more referrals from farther and farther away that will bring in revenue to the city. Commercialization of discoveries, the PFK 158 project I mentioned with ACT is a good example of that. And then there's this concept of productivity. If the local inhabitants don't have to worry about getting good cancer care, they're going to be able to have better productivity locally. If we have a great healthcare system in Louisville, Kentucky, we're going to be able to recruit the most talented individuals to actually work here. And something like a 5% sales tax seems like a small price to pay for all those benefits. So this is my final slide. The guy in the middle there, you probably don't recognize, but his name is Jim Watson. He's known as James W. Watson. And Jim Watson and I were sitting at dinner. I was at a meeting in Cold Spring Harbor, and he was sitting next to me and chatting away, and he's a very eclectic guy. He was the co-discoverer of the structure of DNA in the 1950s and won the Nobel Prize in the early 1960s. And somehow I convinced him to come to Louisville and give a talk. He came and ended up being 3 days over Kentucky Derby in 2011, actually. The way I got him here was, of course, the Kentucky Derby. So he came and he brought his beautiful wife, and we had a great time. And this was right after his talk. He was taking Q&A. He had about 1,500 people in the audience, and someone asked, how do we cure cancer? And it was fortunate there was someone taking notes on all this. And I said the kind of dorky scientist thing, and I talked about, well, we have to come up with drugs that target specific things in cancer, like the PFK 158 thing. But this is what Jim had to say. Jim said, provide more money to research-oriented cancer centers to let them take on low probability, high payoff projects that pharmaceutical giants reflexively turn down. And that's the projects I just told you about, the 3 projects. No pharmaceutical companies were touching any of those. They're not interested in those. They're way too high risk for them. They want to take a compound out of their basement and test it against 5,000 different cancer lines and hope that one of them has an effect. So what Jim was really saying was we need to put more money into these local cancer centers where a lot of innovative research is happening. And then Dr. Miller said something that he said to me almost the first day I got here. Always remember that the human experiment is the most important. We can cure cancer in mice, no problem. You can't cure cancer in humans. We know that. We're 500,000 people in the United States know that. The only way it's going to happen is if we do more clinical trials and move agents into phase 1 clinical trials more rapidly and fund more basic discovery and innovative research. And that costs a lot of money, doing those type of clinical trials. It just does. But I think we're really close to curing cancer. And I think in my lifetime, I'm going to see at least 3 or 4 new types of cancer become chronic illnesses that will no longer kill patients before their normal lifespan is up. So with that, I'll take any questions that you may have.